Okay, we'll go ahead and get started. I'm happy you're all here. This should be fun. Welcome to the inaugural tidybiology meetup. The hope was, and the plan was, to walk you all through the boot camp in order to get everybody on the same page with sort of a very early rudimentary level of understanding with R and RStudio and how to use those tools to import data sets, manipulate them, and visualize them. And that was just the entree. The main course, so to speak, will be this monthly planned tidybiology meetup. The format for this will be pretty relaxed, pretty casual. So you can see there's a little bit of food and drink in the back, so help yourself. We've got Mickey Mouse Clubhouse going on up here in the front, so if ever you want to sort of join Grace and tune out, so feel free. And yeah, so the point here really is to find a community that's interested in learning, continuing ongoing education and learning together, and to also help each other in order to increase our level of data literacy. When I put some of these lectures and these ideas together, I learn. When I explain them to you, I also then realize some new things, and also to build some community. The format of these will be completely fluid and flexible, but one very early idea is to include also a short research presentation, so we're all a little bit more aware of how people are using R in their ongoing research. We have a fantastic work in progress seminar series, so this doesn't intend to overlap or usurp that by any means. But really the focus here is to identify people who are using R and who are using data science in their own research and to get a sense of those things. The very brief introduction, most of you are in my lab, but those of you who are not, my name is Matt Hershey. I'm an associate professor here. I've been at Duke almost 9 years now, and I started down this rabbit hole that I call data science about 2 years ago because I wanted to really develop more robust and reproducible workflows for our own metabolomic and proteomic data sets. Now, I'm a little embarrassed to say 2 years later, we still do not have robust, reproducible workflows for our own omic data sets, but we've learned a lot along the way, and it turns out that's actually one of the things that we're actively working on. So, but what I realized along the way is that there's so much more to data science than just producing a workflow, a script, a series of functions that you can then take your data, run it through, and get a pretty list out the other end. In fact, we're working on projects to try to integrate data sets to help us come up with better hypotheses, or to ask very simple questions of very old data that doesn't make sense or that doesn't coincide with the things that we know. So I will be your fearless leader, but I'd be remiss to not mention we have two fantastic people that I've convinced to come along this journey with me. So I'll let Ali and Akshay just take a minute or two and just introduce themselves. So go ahead.
My biggest motivation is that I want to make this really beautiful reproducible in art. I find this to be the
I'm Akshay. I work in White's lab. We do aging research. We focus on caloric restriction and skeletal muscle. I too started to teach myself R around 2 years ago. Like most people, I started off with alpha data science, which I highly recommend. Yeah, it's a lot of fun.
Okay, so Ali will be giving the short research presentation today. So one of the things I just want to mention is that we're also going to live broadcast these, but then also video record. So if you have friends, colleagues, roommates, parents who really wished they could be here. Hi, Mom. That weren't able to. These will be on YouTube, on the Tidybiology YouTube channel afterwards. Also, if you weren't able to attend our boot camp, all of those lectures are also on the Tidybiology YouTube channel, so you can get caught up anytime. So generally, the format, I think, and again, this is fluid. I'll be asking for your guys' feedback. But the format, I think, will be something going along with a 10-minute introduction. And in this time, we can also share results from a visualization challenge. I'll explain that in just a moment. But we'll just have a short sort of housekeeping, intros, sharing some visualization data sets or outputs that we generated. Then we'll have a very short, maybe like 15-minute research talk from you all, hopefully. This week will be Allie, who will tell us about one of her projects and then how she's using non-targeted metabolomics to gain insight into her project. And then the meat of the meetup is going to really be very task specific. And so I, along with Akshay and Allie, have identified several tasks that we think would be useful for you all to know. I, again, will solicit your feedback because you are the ones who are here. If you have things like, oh, I really wish I knew how to fill in the blank, those would be perfect topics for future meetups. So my hope then is we can go through some code together. You then will have access to this code. And if you want, like today we have non-targeted metabolomic data. If you want and you have a metabolomic data set that you want to use in place of the training data set, fantastic. If you want to take this code home and try to apply some of your own metabolomics data to it later, even better. And again, this is a community that we can all leverage to try to figure out how to help you in your research, to help you in your own sort of training and journey. So the bulk of the meetup is going to be a very specific task. So any questions before we dive into to start?
What platform are we going to be working in that? I wasn't sure, so I've got an R Studio Cloud tab here. I got R on the computer. How do you want to?
So so the question is what sort of infrastructure will we use? You still have, if you joined the boot camp, still have access to RStudio Cloud. You're more than welcome to absolutely use that. If you don't have that, then we can help get you set up with RStudio, just a local install today. Either of those are going to be fine. One of the first things I'm going to show you is how to access this repository, which will then have the code, have the data, have the visualization challenge, have all of these things. If you have problems with that, or problems that I'm going too quickly, or you don't quite understand something, most of you have a bright fluorescent colored sticky note, well, pastel fluorescent colored sticky note. Go ahead and just put that on your computer, and Ali or Akshay will come by and help you with that. So yes, Tim, we're going to start with some infrastructure to get going. Okay, so this is the GitHub page for the Tidybiology meetup. So my lab has a GitHub account. It's called Hershey Lab. And then we have a couple of pinned repositories at the top here. So go ahead and open up a browser and just go to github.com/HersheyLab. Extra bonus points if you spell it correctly on the first try. It's also in bright bold letters right here. So once you're on this page, then what you can do is go to Tidybiology Meetup. So the simplest organization and the simplest way to get started is going to be for you to first be familiar with the structure and the organization of this repository. So this is called a repository or a repo. So this tidybiology-meetup repo then will contain all of the information related to these meetups. If you missed the Tidybiology lecture series, you can see the lecture notes here, slides, and so on. So the YouTube page, which we'll put a link to the YouTube page here, but the YouTube page then references these files. So that's in the Tidybiology lectures. We are in Tidybiology Meetup, so if you click here, then what we'll have is a folder for each month. So right now we're in January 2020, so you'll see that this folder right here, if you click on this folder, you'll see that it has a few subfolders or subdirectories. We will get used to this sort of organization where we have a folder for code, we we have a folder for data, we have a folder for results, and then also in this case, we have a folder for slides, and this is where I put Ali's slides for her talk today. Okay, so code is a bit self-explanatory. This is going to be code. Data, this is where raw data goes. And so this is our CSV file that we'll be working from today. This is results. So we have a few results output, PNGs for some images that we make. So one thing that you can do is if you ever have problems, these RMD files, you can just click on these. And this is all of the code right here. And it's just as simple as cutting and pasting. Right? But that's a little bit unsophisticated. So one thing that we want to do is we want to be able to have you guys simply access this code, and that's going to be to do something that's called clone this repository. All right. So if you are in RStudio, or if you are in RStudio Cloud, then go ahead and get to a page that looks something like this. Right? So you open it up. And just launch RStudio. Does anybody not have RStudio already installed or RStudio Cloud? Okay. So this is what you should see. And we want to go File, and we want to start a new project. That's here. And we're going to create a project, and we're going to click on Version Control, and then click on Git. Okay? So what this is going to do is it's going to take a copy of this repo, and it's going to clone it onto your machine. And we only have to do this setup one time. These setup instructions are also in the tidybiology YouTube channel install instructions video. So if any of this is confusing or you need to go do this later to another, like if you're in RStudio Cloud now and you want to put it in RStudio, you can watch this later, or you can watch that install video. Okay, so back on this page, you see a green button here that says clone or download. If you click on that one time, it will have an address right here that if you click on this little icon, it will copy the address. So click on that one time. Now what we want to do is go back to here, and we're going to clone this repository and just paste in this first URL box. And what will happen is it will paste the URL. It will also make a directory name that is the same name as the project repo, which is tidybiology-meetup. What you want to do is you also want to click Browse here, and you want to put this project wherever you are going to put this project. Okay? So you can put it on your desktop. I don't recommend that. You can put it in some other directory that makes sense. One of the topics that we'll cover in a future meetup will be project organization. So don't worry about that for now. You can just put it in your My R Projects folder or anywhere. Okay? And then as soon as you, if you have another session open, you can say create a new session, but if you don't, then you just say create project. I've already done that. And so you should see now something that looks like this, okay? Where over in this section of your RStudio, you should now start to see a project file right here, README files, and importantly, this directory, which is 2020_1 for January. So remember, if you have questions, go ahead, take that orange sticky, put it up. If you don't, then congratulations. All right, now, you've done this one time. Fantastic. You won't ever have to do that whole cloning again until you have a new project or a new repository that you want to clone. So now every time that you're going to get some new information, you're just going to click on one of these buttons up here. We'll talk about that next time, all right? But now you should all have this repository. And importantly, you can think of this. This is a snapshot or a clone of this repository at this current time. So next month, when I update this and I put a new folder here that's called 2020_02, then you guys will push a button. You will then just download this new snapshot. Okay, so this is how it's going to work. So now you guys should, if you click on this, you can just double check to make sure that if you go to code, you've got some code. If you go to a data, that data file is there. Okay. Now, one of the things that I thought would be fun to do is to have a project that you guys could have to work on, a small project all month long, that then next month, at the end of February, you can come and we can share the results of that project. So this is a visualization challenge, or a visualization a project. So if you don't want to participate, that's fine. You don't have to participate. If you want to go deep and make 17 different visualizations, talk to your PI, make sure that that's okay, right? But otherwise, go nuts. It'll be fun. So there's an activity within the R for Data Science community that's called Tidy Tuesday. So Tidy Tuesday is an activity that we've mentioned before in the boot camp, where on Tuesday a data set is released, and then people essentially have all week to try to take a data set and to turn that into some clever visualization. And it's amazing because you'll see a range of different types of visualization outputs from a single data set. And everybody's using the same data set, and people then will ask different questions, and will look for different associations, and some people will do things as simple as counting. And some people will do things as sophisticated as building sophisticated machine learning models. And you can be anywhere in between, okay? So in here, if you go to 2020_01, and you go to data, you go to code, do this all now. I want you to open up tidybiology viz January RMD. Those HTML files, don't worry about that. You can delete those, we'll remake them. But if you open up this visualization January, this is going to be some early code for you all to participate in this visualization challenge. Okay, so if you guys then just, if you are in RStudio or RStudio Cloud, you might have a little note up here at the top of RStudio that says you haven't installed these packages. If you have that note, just go ahead and click install. If you don't have that note, then you might need to install some packages anyhow. There's a couple of packages here that I like to use that are useful for this data set, or for this visualization. So you can go ahead and try and click run all, and just give it a second. It will download a couple of files. If you do that and it breaks in that first code chunk, then go ahead and put that orange sticker up. You might need to install some packages. Remember, if you want to install some packages, one of my favorite and most useful packages is called Beeper. It's a great package. It beeps when it gets to that line of code. So I always put it at the end, so it will tell me when the code is done. And so if you don't have Beeper, remember the function install.packages parentheses in quotes Beeper will install that. So that's the other manual way to do it. Okay, so for our January visualization challenge, what we'll do is try to not touch the cables. What we'll do is we'll use a data set that's called Prism. If you go to a web page right now and you go to actually DepMap, which is a project that the Broad runs, you can see that there's a lot of different great data sets that the Broad Institute has made publicly available. One of them is called Prism. That's here. And Prism is a powerful approach to rapidly screen thousands of drugs across hundreds of human cancer models on an unprecedented scale. So right now, if you ran that code, you now have the Prism data inside your RStudio environment. It should be called Prism, and you should also have Prism_meta. So Prism is the Prism file, and Prism meta is the metadata that goes along with it. So if we just do this quickly together, and we view Prism, we can see that we have 578 observations across 4,687 variables. And what happens here is we've got a cell line, and we've got a drug, and we have a log2 fold change for how that cell responded to that drug. Okay? So one of the things when you do an exploratory data analysis or any visualization challenge, the first step is an exploratory data analysis. You really want to understand the data. This is why I point you to the DepMap, because this is where the raw data come from. You can read actually the paper if you'd like. You can read about how they generated their data and so on. It's really a quite clever approach. But now what we have here is, let's see, we said 578 cell lines, and they've treated 578 cell lines with 4,600 different compounds. Well, that's really remarkable, and we have log2 fold change values. If we come down here to Prism Meta, what we have is some metadata about the compounds that we treated. So we see that we have a column name, we have an ID, and that's the Broad's ID for their compound. Then the name of the compound, the dose, another ID column, mode of action here, immunostimulant, estrogen receptor agonist, etc. Target, if it's known, in this case, first one's not known. Here we have GPR1, and so on. Disease area, sometimes we have a disease that's associated with this compound, cardiology, endocrinology, rheumatology, and so on. Indication, SMILES, that's a chemical compound code for a compound, and then also what phase it's in. So the idea would be then to take the PRISM data, to take some of the metadata, to join those together, and to try to do some clever visualizations. So you can see that we imported it here. We've got all of this information. And then two exploratory things that I wanted to know. First was about the compound dose. So we have a compound dose in here right now. And I just did a quick GM histogram. So this first, seriously, 2 lines of code, is I took the PRISM metadata and I did a GM histogram looking at dose. And what you can see is that essentially everybody got 2.5 except for there's a couple out here, one here, and then maybe this one got much, much lower because of some toxicity. But you can see that this 2.5, and if you go to the metadata file, you can read that this refers to micromolar. So in this case, all of these cells got 2.5 micromolar except for a couple of outliers. So maybe you could ask a question, well, what are those outliers? Who got more, who got less, and so on. The second thing I wanted to know, again, just quickly, is what's the range of log2 fold change values? So we took all of these drugs, we put them on all of these cells, we say, how did it change? So we then come here, and this is, again, just a really quick histogram. So 0 would be no log2 fold change. These guys might have grown a little bit better, and you can see that these were like super toxic, and these like die a little bit, and so on. So this is sort of, again, the shape of the data, the sense of the data. So if you're interested and you're curious, then the challenge for the month is going to be to try to do some visualization. We can take some of the metadata of these cell lines. We can then do a left join with the data from the log2 fold change, and we can ask about maybe some associations about toxicity, or we can ask some associations about what are the drugs that grew better or that made the cells grow better, and so on. So if you're up for it and you want to try it, this will be a fun data set to explore. One thing that I'll just mention, we didn't mention this in the boot camp, but one of the things that you want to do is to take your data and to convert it long. So one of the first things that you want to do if you have some data that looks like this is you want to do a pivot longer. We can talk about this maybe next week, or in fact, I think that's one of the proposed topics, maybe not for next week. But this line of code here I gave you will take then the PRISM, slice. This is what I do sometimes just to practice with the data to make sure my code works. But then you can take pivot longer, and you can then convert the names to drug, the values to log2 fold change, and then you get a file that I called PRISM long. And this is the file, PRISM long, that then has X1 is our cell line name. The drug is the drug name, the Broad's ID plus its drug name, and then here's a log2 fold change. And this is the data format. This is the tidy data for plotting. So what I wanted to do in this just first part was really just introduce you to the concept of a visualization challenge. So some of you asked for homework, believe it or not, some ongoing, something that you could work on throughout the month. So then I think at the very beginning of our next month, what we'll do is when I send out the reminder email, I'll also ask you to email me any images that you made from this challenge. And don't forget to put your sort of caption on the bottom of your ggplot so we can attribute it to you. But I'll ask you just to send it to me, and then we can put them up there and we can see who did what. I thought it would be interesting because I've always found it useful to see both the beautiful plots that Ali is striving to make, but then also how people think about it. Like, oh, I never thought about looking at the relationship between whatever. So I find that that's the most useful. So if you have questions about the challenge throughout the month, just go ahead and email. But now, for the rest of the time, what I thought we would do is Ali will give us a short overview of one of her projects, and then we can, I'll dive into reviewing the code that recapitulates some of her graphs. So, any other questions before we get going? About the visualization challenge, about the colonial repository, about the data? Okay. Well, then Ali's going to talk about metabolomics. Ali.
Hey, so once again, I'm Ali. I'm a postdoc in Matt's lab. And as many of you may or may not know, Matt's lab has dealt a lot with sirtuins, particularly sirtuins 3, 4, and 5, which are mitochondrial sirtuins. And I'm going to talk to you a little bit about a project I have involving SIRT5. So sirtuin 5 is a deacylating enzyme that removes these acyl modifications from substrates. Its main targets are malonyl, glutarol, and succinyl post-translational modifications. And while we don't understand the biological importance of SIRT5 just yet, it's been implicated in a number of metabolic processes, including the TCA, fatty acid oxidation, amino acid metabolism, ROS response, urea cycle, and glycolysis. However, I actually started working on SIRT5 in grad school and came to SIRT5 through a completely different angle. And that is, I was looking for substrates of this complex called the SCF cyclin F, which is a ubiquitin E3 ligase. So what this complex does is target SIRT5 for ubiquitination and degradation in a cell cycle dependent manner. So as I go through what our model here is, I'm going to give you a little snippet of data for each little part, okay? So for this, I'll go ahead and show you that what I found was that SIRT5 stability is increased in the absence of cyclin F. So here we have an experiment where I took HeLa cells treated with cyclohexamide, which prevents the translation of new protein from mRNA, and I measured the half-life of SIRT5. So here it's between 4 and 6 hours, which fit with previously published data. However, when cyclin F is knocked out, we see that SIRT5 half-life is no longer measurable within the time frame of this experiment. So that's part of what convinced us that SIRT5 is a target of this ligase. What's particularly unique about this ubiquitin E3 ligase is that all of its substrates to date have known roles in cell cycle progression. However, we don't know of any roles for SIRT5 in cell cycle progression. So not knowing all that Matt's lab knew about metabolism and knowing what we did know about cyclin F, we thought in my previous lab, SIRT5 is going to affect cell cycle. So this was our hypothesis, was that SIRT5 was going to affect cell cycle, and what we found particularly, SIRT5 inhibits the G1 to S phase transition. So this data, here's a quick glimpse of that, is in a regular 293T wild type cell. We have here a little diagram of the cell cycle distribution. So how to read this is this first peak is the G1, G0 peak. S phase here, that's where cells replicate their DNA. And then G2 and M, so that's where cells divide into 2 daughter cells. And this is the distribution of a normal wild type 293T cell. However, in the absence of SIRT5, what we see is this big decrease in the G1, G0 peak. And that's consistently a 15 to 20% decrease in the G1 population. So this is what led us to believe that SIRT5 is inhibiting this G1 to S transition in cells. Furthermore, we thought not only is it inhibiting this transition, but it could be promoting the G0 entry. G0 is a part of the cell cycle where cells have monitored their surrounding environment and determined it is unsafe for them to replicate through another cell cycle. So instead, they go into this reversible phase called G0 or quiescence and wait until the correct nutrients or space or whatever is available for them to re-enter the cell cycle. We can induce cell culture cells to go into this state not all, but some types of cell culture cells. And I performed an experiment where I took normal human fibroblasts and I forced them into G0 by either contact inhibition, which means I let the plate get confluent and the cells don't have any more room, or serum starvation. And what you see is that cyclin F goes down in these conditions, but SIRT5 goes up. So this suggests SIRT5 could play a role in either helping establish or maintain quiescence. But we still have one major outstanding question, and that is, how is SIRT5 influencing cell cycle progression? And so at this point, I brought my work here to work with Matt, and a previous postdoc in the lab had data showing that SIRT5 cells have reduced amino acid catabolism. Specifically, the SIRT5 knockout cells have high levels of almost all of the amino acids compared to the wild type cells. Furthermore, they have a reduced ability to oxidize or leucine and lysine, meaning they're not breaking down these amino acids as efficiently as wild type cells. So we thought perhaps SIRT5 is regulating a metabolic pathway that's influencing cell cycle, particularly amino acid catabolism. So we already know that a major sensor of cell cycle progression is nutrient availability, or a major contributor to that decision to divide. So we performed non-targeted metabolomics on SIRT5 CRISPR wild type and knockout cells in a few different conditions. First of all, we took wild type and knockout cells that had been fed regular media. We then fasted some cells for 2 hours in a salt solution. These have no serum, no glucose, no amino acids, so there's no metabolic fuel for these cells. And then we also refed cells after a 2-hour starvation with regular DMEM. So I took these cells, performed non-targeted metabolomics, and analyzed the wild type versus knockout of each of these. And I'll zoom in. This is what we'll be looking at today. The biggest differences we saw were in the fed state. And everything I've highlighted in pink here is actually a metabolite that's found in amino acid metabolism. As we look at the fed state and the fasted state and the refed state, we see many of the same things going up or down. So these are mostly all involved in amino acid metabolism, supporting some of the data from previous members of the lab. So that's where I am with that, and I didn't want to make it too long, so I'll go ahead and thank everybody in the lab and give it back to Matt.
Any questions?
Yeah.
Have you checked the expression of SIRT5 during the course of proliferation?
Yeah. So you'll notice on my here. Maybe I'll get back to on this slide here, you'll notice I have on the bottom, SIRT5 protein levels must dip below a certain threshold to enter S phase. So they're never actually completely gone. I didn't go into the finer points of the fact that SIRT5 is a mitochondrial sirtuin, meaning it can enter the mitochondria, but not all of it's there. But cyclin F cannot enter the mitochondria. So it's really can't be targeting the entire pool of SIRT5, which is part of what makes it a little bit more difficult to tease out.
Maybe so how are you measuring the relative distribution in what phase it's all in?
The flow cytometry, those are simple fixed, where we fix cells and stain them with PI and just measure the distribution based on the, it's basically based on the size of the nucleus. I've also done EDU incorporation, which tells us how many of the cells are actively in S phase, because that method I showed you there is a little bit vague in some senses, but they all point to less of the cells are in G1, more of the cells are in S, G2, and M.
Okay. Thanks, Alan. So, now the reason you all really came, show me the code, right? Calm down, Tim. This. So, if you close that the January Viz RMD and open up the other RMD file in the code directory, you'll see a MBX, metabolomics, volcano _ cert5. So this is, again, this is some data that you should have also in the data folder if you have some other code that you would want to analyze. You can follow along if you'd like. But the plan now for the next 20 minutes or so is for me to walk you through this code and show you, sort of explain step by step what each line is doing and the rationale for doing it, and to have you guys ask questions along the way. So, any questions before we get started? Okay, we'll go ahead and go then. Okay, so this is our Markdown document. So the idea here is that this, again, can integrate some code, some plain text, and to generate a reproducible report. So here, I have a title, an author, a date. So in your date, you can actually do a backtick R, which is the code to run a piece of R code. You can do this in this what's called a YAML header as well. So you can put the system date. So if I knit this file, what will happen is it will include today's date. I'll just show you quickly. It should just take a few seconds to do this. And then we have two different output types. So one is a HTML document, one is an HTML notebook. This essentially will allow us to view the output here, and this is going to just open it up in a web browser. But you can see, like, here's my title, author, here's today's date, and then this is kind of nice because it has a table of contents here, and then we go on with the code. So all of that's happening in this YAML header. So
what's the difference between an HTML document and a notebook?
I knew someone was going to ask that. I should have looked it up. No, so I think what's happening is when we preview this notebook and we knit to an HTML, so if I take out, let's say, this one right here, okay, and I save it, and I say knit to the HTML, it will make my HTML document. What happens is I think, so the short answer is I don't know. I don't know exactly the nuances of the difference. Perhaps I can report back to you next week. But the document will make an HTML page as an output, just like it will make a Word document as an output or a PDF file as an output and so on. The HTML notebook, I think, is going to include some different sort of formatting. So you can see, like, I deleted that and we still have the exact same options here. So, but I can look and see exactly what is accompanied with the notebook. What I envision is it's something having to do with the number and types of files that are included with it. Okay, so then I always get in the habit of trying to write a short introduction here. So this is our Markdown notebook from the Hershey lab to generate a volcano plot from non-targeted metabolomic data. So just again, to remind myself to intentionally declare what the point is, what you're doing. And then our first setup chunk is going to load libraries. You can call it setup if you want to. I didn't in this case. And I'm going to include a handful of libraries. So one is a tidyverse. So this is going to be our base packages that we'll use to manipulate our data. Janitor is a package that cleans column names quite well and has a few other sort of helpful functions to make it easy to clean data. Viridis is a color palette that I like to use. It's a colorblind friendly color palette. So if you use colors like red and green, the 10+% of the male population that's colorblind won't be able to see this, including a former postdoc in my lab. So we try to be aware of accessibility, and Viridis is a good default way to do this. They're also pretty aesthetically pleasing color palettes. ggRepel is a package that will make labels and ggplots that repel away from the points. So when we put our plots, when we have our volcano plot and we want to say which point is which, we can have a smart way to label this without blocking all of our underlying points. Broom is a great package that's under development by our studio and several developers that will then take the statistical transformations of several base R functions and convert them into a tidy format. So oftentimes when you have a function, let's say t-test, that's one that we're going to use today, the output of the base R t-test is a mess. And what we want to do is try to adhere to some of these tidy principles, tidy data, and put things back into a data frame. And so Broom is a package that sweeps up behind this and creates a nice tidy statistical output. And then Beeper, all it does is beep. But it tells me when the sort of everything's done running. So clear environment, I comment this out because I don't want to run it by default, but I can go like this and make my environment empty so that I can make sure that I'm starting from scratch just like you are. Okay, and then finally, you can also set theme. This is going to be a way to set the theme of the global environment here. So if you have something that you like to do often, you can put that sort of in the setup chunk here so that all of the plots that you generate will all have the same theme, which then means you don't need to call that theme every single time. Okay? So this is my load libraries, this is my setup. Now, quite practically, when I'm going through this, I'll load a library, I'll say tidyverse, and then I'll start doing some things down here, and then I'll come back and say, oh, I need janitor, and I'll come here. So I would encourage you all to get in the habit of calling libraries all at once in the front, because then you know which ones you need for the entire analysis. You don't have to. The only requirement is to load the library before calling a function of that library. But I think it's good coding practice to call them all up front here so you know which ones that you need. Okay, so you all with me? One chunk down. All right. Now, next, we need to get the raw data in. So this pulls the data from the master Excel spreadsheet, or in this case, it's actually a CSV file. All right, so we load the data set. I like having a code chunk here where I load the data set. And the first thing I'm going to do here is read CSV. So that's my function that will read my CSV in. Now, there's a clever little package here, or a library called here. Now, I could load my library up here. I could call here up here. Another thing that you can do is you can actually call a specific function from a specific package. So my function is called here. My package also happens to be called here. So when I write here, it means I don't need to call my function up here. I just wanted to show you that just as an alternative. But anyhow, here is so let's actually go ahead. You guys can all do this. Let's type in library here. And go ahead and just run that. So you can run that command or that line of code by hitting command return. And then you can come down to the console here and type here parentheses. And what it will do is it's conceptually equivalent to git working directory. So we'll tell you the working directory of your project in this case. So my project is users, me, Dropbox Duke, projects, tidybiology meetup. Now, here is a nice little light package that allows me to quite simply refer to everything relative to here. So here is going to be the sort of the high level project, which is tidybiology meetup. So then you don't have to remember that there's a slash go before or after. You don't have to remember exactly like where I am. You can just use this very simple syntax of writing like one folder, the nested folder, and then the file that you want. Okay, so I like here as a nice a way to refer to directories because then I always can't remember if I need a slash, a leading slash, or a trailing slash, and so on when I'm calling these. And then also, if I hard code where my data are, like if I write out that whole path, and then Keith tries to run that on his computer, it breaks because he doesn't have that path. So this is a nice way to make this code more accessible to other people. All they need to do is have a folder called this and a folder called this, and then these data in the folder. So even if Keith had these data, but it wasn't in the right spot, it would break. But now it's much easier for Keith to put those data in the right spot, and then he doesn't need to go back and hard code his path because the path is relative to his directory. All right? So that's a nice little a package that you should know about. Okay. Column names equals true, trim white space is true, and then this clean names function. So this is going to import my data, and then so I go like this, and you can see that I've got some data here, I've got a metabolite, and I've got some chemical formulas, and then some like terribly formatted column names. So if I say clean column names, that's that package from Janitor. Then what I have is a very slick way to make everything, it takes periods, converts them to underscores, it makes everything lowercase, it basically converts it all of these column names to like a standardized way. So it's a much easier thing to do than going through and manually changing, like manually cleaning every single column, which is going to become important if you have hundreds and hundreds of columns. And then there's another function you can do here called remove empty. I put here just in case, hard to know if there are empties. If you put a CSV file in and there's a bunch of empty columns at the end of your CSV or Excel file, then you have basically an empty vector that then causes all sorts of problems later on. So one thing you can easily do is basically trim to remove empty. Do you want to remove empty rows or columns or both? In this case, remember this C combines the two variables. So in this case, I want to combine rows and columns as my arguments for remove empty. And in that way, I can remove them. And then what I want to do is I select metabolite and starts with. So this data set, it turns out, has a bunch of other information. And so let's see if it'll come up here. So like this and this, those are empties. So I need to get rid of empties, but then I want to basically make sure that I'm only getting metabolite. And in this case, this is very specific to this data set. The way that I receive these data was having an II at the start. So then I say starts with II, and there we go. So if I run this whole thing, then what I do is I basically have everything clean. And actually, let's just go like this. We can run this whole chunk. There we go. So we have a metabolite, and we have all of this. So, everybody there?
Nope.
Okay.
So, this was the first introduction. You underestimate the time it takes some of us to get packages in here. So I'm still trying to load the packages that I don't have on mine.
Okay.
I'll get there, but before I get too far behind, I know I can do this on my own time tonight, but if I can follow along, that'd be better.
Okay, sure. We'll take 1 second. So, so RStudio will usually detect the packages that are called, and if you don't have them installed, it will install them for you, or it will ask you, would you like to install them? It won't install them for you. It will just say, would you like to install these packages?
Mine's not, I'm just getting errors. It says, oh, can't move past, here it is, because that's not there.
Okay.
So,
Yeah, I don't think mine's ever asked me if I'd like to install it. You just have to install it.
Okay.
Easy way to install that.
Anybody else have any questions so far? Okay, nope, that's fine. So now we have data, we call it data raw, and it's in here, we can view it, we look at with the head function, data raw and equals 10, and we can see that we've got metabolites, and we've got our conditions here, and so on. All right, now, the problem, and we see this problem oftentimes when we get data, is, I keep running into the cord, that's the problem. So the problem oftentimes when we get data is that it's not in a tidy format. So the tidy format, if you recall, is the maybe? So the tidy format is one observation per row and one variable per column. You can have that. So let's go like this. So what we have, remember, so you have an observation, and that's one of your samples, and your then.
This table doesn't do that.
Okay. All right. So if you have, whoa, I'll save that for next time. Okay. So we have an observation and then a measurement, right? But what we have are all of our metabolites, which are measurements in rows. In this case, our observations are in the columns, and that's backwards. So we need to do a quick transpose. There are several ways to do this. I chose the most complicated way, but it's the most verbose way, okay? And that's why I list it for you here. So if we have data raw, we can make this longer, and this is the command that we use to get data ready for plotting. So what we can do is pivot longer, and this then says I want all my columns except for metabolite. And we want to convert this because remember, data raw looks like this. So we want to convert, leave metabolite alone, but we want names to be a sample and then values to be a value. And what happens now is we have our sample here and our value here. And then we can pivot wider and we can say IDs, ID columns equals sample, names from, values from. So these are two really useful functions that will basically take something that's square and turn it into a rectangle. And then what we do is we take that rectangle and convert it back to a square. So this pivot language is a newer language. It was previously spread and gathered, but this pivot language is actually from Excel and from SQL and from a few other sources where we will pivot the data to make them long and then wide. And if we change the variable, then we can essentially transpose. And when we do that, let's run this line, and then we'll run this line. We can see now we have one observation per row here and one variable per column. Okay, so now we have something that looks like it could be a tidy format of our data. Okay, now this is nice, this is useful, but what we want to do is we want to now add some information. We want to add factor level group IDs as a new column. So we need to group these in some meaningful way. We just have a sample, and I know because I can read here that this is like a wild type. So let me back up. This sample is a metabolomic, the metabolomic data set that Ali presented earlier. So we have now cells that are wild type or knockout, and these cells then are either in the fed, starved, or refed state. So fed is just cells growing in a dish. Starve is where you wash them, take the media off for like an hour, and refed is you put this back on, let's say for an hour. Okay? So we want to add some information back to this beyond having just the sample name. So this is seemingly long and complicated, but we can read through this quite quickly. We'll do this together. So if we have our cert5 data set, recall that mutate is a way to either take a current column and change it in somehow, or we can add a brand new column that doesn't exist yet. All right? So we're going to mutate our cert5 data set, and we want a new column called group ID, because that is not in this data frame yet. So group ID is going to equal. Now, what we want to do... Yeah, you can stand up here with me. Yeah. Here, come here. Okay, so what we want to do is we want to then take our group ID, and we want to now call the group ID something special based on some programmatic way of referring to this sample here. So Casewin, so I previously had something here that was called if-else. So if-else statements are quite useful, but if-else statements are, you can stand here. If-else statements are going to be useful if the sample then contains this thing, then I'm going to do this. So if it does this, I do this. If it doesn't do that, that's where the else comes in. Okay? So if Gray stands next to me, then I feel happy, else I feel sad. Okay? Now, this becomes complicated if you have several if-else statements all strung together. So the Casewin is a function that you can learn about by coming over here, going to help, pasting in Casewin. A generalized vectorized if, right? So this function allows you to basically string multiple if-else statements together. So the way that we read this is, it's looking for true-false here, remember. So string to text within sample. So these are kind of silly names because they got a little bit mixed up, where it's like the wild and the type is now separated, whereas the wild type here is not. So I need to come up with something that is unique to the wild type fed, and that's where tfed comes in. So this basically says when I detect within sample this set of characters, then I'm going to call it this. If I detect this, then I call it this. If I detect this and this and this and this and this, and if I don't detect any of these, then I'm just going to call it this one, which is the one that's left over. So this is very specific for this example because I need to then refer to my samples here. But if I do that, and I go like this, actually, I'm going to just run the whole thing because then I put them in the front. So select sample group ID, everything. So then what happens, I go from sample, I now have this nice group ID where I have wild type fed and wild type starved. All right? Yeah?
So in this context, then, do you have to make it another column, or can you mutate the names in that column without affecting the first several characters, which obviously you can't universally mutate?
I could, but this is my like original. So the question was, could I just have mutated to change the information in sample? And yes, I can. But the reason I didn't is twofold. The first is because I want to preserve this original raw information. Right? So if I start changing things, then it's hard to go back and double check. The second thing is this has a lot of different types of information. It has my genotype, it has my status, whether I'm fed or fasted or so on, and it also has a replicate number. And that might be important because like keeping a replicate number would be important for something. So if I just then start changing this, then I start losing information. So really what I'm going to do next is I'm going to start pulling more information out of it. Okay. So next I'm going to log transform. So this is something that we oftentimes do. We transform the data to sort of normalize and put it all on the same scale. So in this case, I take my cert5 data set, which I just made. And this is a nice little function. This is called map. This is in the per package, P-U-R-R. I think that's it. Maybe there's 3 R's. Anyhow, this is going to run a function, but this is a little bit like a mutate if. This map if says run my function if it is numeric. So I'm going to take a dot. This just means I'm going to take my information that I passed to it. So I'm going to take my cert5. So I'm going to take all of this. I'm going to then see if it's numeric, and if it is, I'm going to apply a function to it, and this function is a log2 transformation, and that is within the base R package. So when I do that, I see something like this. That's taken a long time. So I see something like this, but really what I want to do is I want to make it a tibble again, so it's back in the same format that I gave it, because this log2 is going to not do that. So then I now have my information, I have my group ID, and now I have a log2 transform value. So I had a data frame, cert5, and I basically can convert it to a log2 transform data frame that matches perfectly.
So Matt, this is what bogs me down a lot. How would you know that you need that function? And I mean, there should be a simpler way just to do a log2 transform. But you knew, or it seems like you wanted to do more with it, so you do something different?
Well,
or is that the simplest way in R to do a map if? I mean,
so think of it this way. There's 3 different, so the question is, how did I know to want to do that, right?
Yeah.
So where's my head at? So here's the thing. There are 3 different key types of objects that you could potentially log2 transform. You can log2 transform a number.
Yep.
Okay? And we can even just do something like this. What's your favorite number?
Yeah, exactly. The 7.
Okay.
Yeah.
Boom.
Yeah.
All right. I can go home now. Okay. Now, if we have an object, and we have, whoa. Okay. Don't push on it. Now, we have another number. What's your second favorite number?
Yeah, 13.
Okay. So now we have a vector, okay,
Yeah.
which is going to be probably like a type numeric or type double. And then what we can do is we can log to Tim,
Right.
and we get something like that,
Right.
okay? So the third type would be like a matrix, or a fancy matrix would be a data frame, or even a fancier data frame would be a tibble, okay?
Yeah.
But in your data frame, which you can think of like, again, we've got 2 vectors right next to each other. What we would want to do is we want to then ask, like, do you want to log2 transform every single number? Because, again, the way to think of a data frame is you've got a vector that's too long in this case, but you've got another vector that's too long right next to it, and another vector. And so the map function is going to then work on vector 1, and then go to vector 2, and then go to vector 3. So if I had a data frame that was full of numbers only, then I would just map. And map is the function that's going to say, take your transformation, log2, and apply it to every single vector in this data frame. But I have something that is not numeric. I have this brand new group ID and sample that I made. Yeah, you can have another one. So you would then want to, what you could do, another way would be cert5 and then select if is numeric and then map dot comma log2. That'd be another totally reasonable way to do it. It's one less line of code because we have a map if, which is sort of a helper for it. Okay? So map is the way that you're going to then take a transformation and apply it to a group of vectors, which is just another way to think of a data frame. You with me?
Yeah.
Okay.
I wouldn't have come up with that.
Yeah. Yeah. Me either. Yeah.
Can I just add something? So one way to do it is like you could do it variable by variable, but then if you find that you have like more than 3 or 4, that's when you should probably use some kind of function. And so the map functions in the Perl package are great for that. But there are, you could use these apply functions that come with base out, or you could write your own like for function. But it's just map is within the tidyverse. So I would Google, I'm doing this. How do I do the same operation for multiple columns in tidyverse? And Google will take me to the Perl package.
Okay.
And then
Yeah.
That's helpful. Thank you.
Yep. Yeah. So there are several ways to do anything, right, in R. And so you could have done a for loop that looped along each of these vectors in your data frame. You could have done an lapply, which is the base R. You could but map was specifically designed to apply a function in a serial way across a data frame. All right. Now, so we have log2 transformed. Okay, that's nice. We'll go moderately fast. I wanted to try to keep these meetups about an hour, so I don't want to keep you guys too long after. But really what we want to do is we want to find a mean, and we want to then compare this to a mean of a value, and then we want to compare all of our values to it. So we have like a wild type fed, and if we wanted to then compare something like knockout fed to wild type fed, we want the mean of each of these, and then we would want to compare it to the mean to again, another sort of scaling step to really say how much different is this. So the way that I chose to approach this, again, not the only way to do it, but the way that I did this was to calculate a mean and then normalize each, essentially each column to that mean. Okay, so this is, I tried to comment this. So I'm going to make a flat group A in this case, which is a vector that I operate the mean function on. Okay, so let's look at this. So I have my cert5 log. That's what I just made. And then I'm going to filter, and I'm going to filter group ID equals wild type fed. So when I do that, it gets rid of everything. Oh, let's go like this. Oh, let's make this one totally. Okay, now go here. And what I have is now a very short little wild type fed only data frame. Next, what I'm going to do is I'm going to do a summarize if, okay, so this is another sort of helper. So summarize if is numeric, I want to calculate the mean. So if I come down here, the way to think of it is not numeric, not going to calculate the mean. Not numeric, not going to calculate the mean. This is numeric, so I'm going to calculate the mean of these three, and I'm going to have one value. Is numeric, going to calculate the mean and have a value. So I'm going to go through each of these columns now and calculate the mean. So when I do that, I'm going to end up with something like this. Okay, so single value for each of these. And then I'm going to flatten it. Okay, so then what it leaves me is a single vector. So instead of being a data frame now, it's just it's flat, so it's just a long vector that corresponds to each of these. And then what I'm going to do next is I'm going to mutate all values by dividing by the mean of the control. And then I need to rebind it to the group ID because, again, I got rid of my group ID here because it's not numeric. So I take this, and then I select if it's numeric. Okay, I've done that. And here's another map function. But in this case, this is a little bit more complicated because what I want to do is I want to map. So let me take this one, and so you can see what I'm looking at. So this is what I have now. And what I want to do is I essentially want to divide each of these numbers by the mean of my wild type group that I just calculated. Is that intuitive? Everybody with me so far? Who's not with me so far? Because the intuition around this should be pretty like solid. Okay, so we've got all of our values. We calculated the mean of one of those groups, and we're then going to divide by the mean. But remember, this is log 2 space, so it's really subtract. Right? But it's essentially going to be a relative value for each of these, and the relative is relative to wild type. Now, what I want to do essentially is I want to take this number here, and I want to divide by the mean. And this one, or subtract. This one, relative to the mean. This one, relative to the mean, and so on. So this is a function called map2 because I'm actually providing two different arguments. I'm taking the value, so this dot is the value here, and then I'm going to give it a second value, which is this object that I just made right here, this log mean control, that flat thing. And then I'm going to basically take dot x minus dot y. All right, so I'm going to take my x, which is this, and I'm going to subtract y, which is this. And to subtract because it's log space. So when I do all of that, I'm almost there. Bless you. Now I have a bunch of numbers, right? So I'm doing this for each of the, you can kind of see as this goes along. I've got a metabolite, and then essentially it's a subtraction for each of these values. But then I want to bind it back to my columns. And then I want to take a look at it. And what I hopefully have is a value here. And now this is not the log2 transform space, but it's our metabolite, that then each value is relative to the mean of our wild type. No, we're going to be done in just 1 minute, okay? All right, so that's what I have in all of this data set. But now I'm ready to start exploring it, almost. So let's see here. So calculate the average. Let's see, so I can then, did I do that one yet? So then I take my log relative that I just made, and then I'm going to calculate the average group by each of these IDs. So if you remember from the boot camp, you can take a thing that you just made that kind of looks like this. We're going to group by ID, and we're going to take the mean of each of these. And then, this is summarize if, is numeric, function mean. And now what we have is a group that's collapsed, and I have the mean relative value of each of these. So then I need to add my genotype and some condition factors back in. So remember, now I have this piece here, and I'm going to add some information. So I'm going to take this, and then I'm going to mutate. I'm going to give it an ID, or a column called genotype ID, and then it's going to be one of these string detect. I don't have very many, so I just did an if-else here. So if it contains wild type, then give it a genotype ID of wild type, else give it knockout. Same with this one. If status ID, so if this is refed, then give it refed. If it contains starved, then give it starved. Otherwise, give it fed. And so when I do these three functions, or these three operations, then what I have down here, this is just a reorder. Then what I have here, so we can just see it, is we've got a group. Now we've separated this out. We've got a genotype, a status, and an average value of the relative abundance of a metabolite. Okay. Now we're almost to the plotting. So we want to make this long for plotting. So we say, I'm going to take this, and I'm going to make a data frame that's ready for plotting. I'm going to say cert5 log plot. So I'm going to pivot. And this actually, I had to go back and figure out the simplest way to do this. But there are some helpers here. And the reason that I gave the names like I did, like status ID, genotype ID, and so on, is because then I can say select all of the columns that don't end with ID. So this little dash on the front means don't. So ends with ID. So that means it's going to get all of these columns again, basically all of these guys. And then it's going to arrange. So I go like this, and we end up with this sort of arranged value, and it's going to be arranged by our metabolite and so on. And then I make these a couple of factors. So this is as factor because I want to use this for my plotting later on. And then finally, I get to do some exploratory data analysis. Okay, so before I do that, I went through this quickly, but the idea is to give you this code so you can play with it, you can explore it, and so on. But who doesn't follow conceptually of where we are right now? Yeah.
What's the as factor? I was up all the way until the as factor.
Okay, so we've got some different types of data here. We need to call it specifically a factor if we want to use that in plotting later on. So I'll show you right now how we can use that factor for plotting. Okay, so we're going to take this data frame. We're going to filter for our knockout fed here. And then what we can do is if we go like this, we end up with a actually, if we get rid of this here, I'll show you, and we plot it, it's going to be kind of a mess. So it'd be nice as if this was like kind of ranked in order. So what happens is we can mutate metabolite, which is really just changing our metabolite column, and then this FCT reorder. So FORCATS is a package that basically deals with all things factors. So FCT means factor reorder. So we can reorder metabolite if metabolite's a factor based on its value of its normalized control. So when we go like this now, we can take a quick peek. We can say, okay, these are the values of my, in this case, it's my knockout fed cells based on the value of the normalized control. Well, FORCATS doesn't work if you have a data type that's not a factor. So in order for it to work, we say, this is a factor data type, so then I can reorder these factors. All right, so this is kind of a nice gut check. It gives us a sense of the data. It gives us like, oh, these are metabolites at the bottom. I never would want to go and like try to make this pretty and see what they are or anything. I just want a sense of like, what's the range of the data and how is it shaped and so on. Okay, so this is nice. This is like our first step of our volcano plot. So our volcano plot of where we're trying to get to is the log fold change difference compared to some statistical value. So this is our log fold change difference. This is the relative difference in log space of our knockout fed versus our wild type fed. But what we want to do next is we want to calculate p-values and then correct for FDR. So we come back to this original data frame, the cert5, and then we're going to filter group ID as wild type fed or group ID as knockout fed, and then select everything except for sample, and then make it long, and then group by metabolite. So then this is, Tim, where it's going to get like super messy, and we're not going to really get into the details here. But what we end up doing at this step, okay, is we end up now with these groups of metabolites. Oh, no, that's not going to work. So we're going to nest it. So groups of metabolites that are now nested. All right, and what in the heck is nesting? That's going to be a sort of lecture for another day. But essentially, the way that you can think of it is it's going to take a group of metabolites, in this case, it's all of the metabolites of hypotaurine, and it's going to condense them into a list. And then it's going to take all of these metabolites and condense those to a list. So it's going to nest these, okay? You could think of like a nested folder structure or those little sort of stacking dolls or whatever, right? So pick your favorite nesting analogy. It's going to nest them into a list because this is where our statistics come in. We can then perform some test on this list, and then we unnest it to get it back out. All right. Now, this took me more than a few hours to figure out, but essentially what we end up with at the end is if we look at cert5 _p, we end up with a metabolite and a p-value. And then it's one simple line to make an FDR-corrected q value. So we take cert5p, mutate, q value equals p adjust, which is a function. What do we want to adjust? We want to adjust p-value. How do we want to do it? FDR. So we do that, and we end up now with a simple column where we have a separate data frame, which is cert5_p, with metabolite, a p-value, and a q-value associated with it. Okay? Now, let me just run all that to make sure that it's fine. Okay. Then what we do, last thing, is we want to join our p-value and our log fold change tables. So we can combine them together so we can get them ready for plotting. So this is a filter and a left join. And it says column metabolite joining a factor and a character vector because I didn't convert that other one to a factor. So this is coercing it back into a character. That's a fine warning to see. And then we make a final plot. So if we start with this plot here, what we end up with is a lot of different points. Okay, let me just do this. So we end up with a lot of different points. Now, if we tried to label all of these, it would be a total mess. So what I did is I made a separate data frame that my labels use, and that's right here. So I make this and I call it significant. And I say cert5 plot and then filter if my normalized control is less than this or if it's more than that, and then filter for q value is less than that, and that's my significant. And then I come down here and I've got my plot. I've got some horizontal and vertical lines to help orient me. I've got my geom point for normalized control, negative log of the p-value, and then my geom label repel. Here I call it different data frame, and this is my significant, which I generated here. All right, scale for viridis. That's going to then make the colors, some labels, theme, make it pretty, and we end up with something that looks like that. So this is a log, negative log 10, FDR corrected p-value, and then we have relative fold change log 2 compared to our control. So then I give it a title, I give it a subtitle, I have a legend over here. Now the data visualization aficionados would say, well, this color actually doesn't convey much more information because I have color corresponding to log 2 fold change, but log 2 fold change is also on this axis. But that's okay because it's prettier. But we really have a few more. We have size and we have color that we could encode some more information here. But anyhow, we can set those thresholds, and then we end up with a plot that looks something like this. We can then also check the top and the bottom, and we can then say, here's our list. We come over here of the metabolites that are the most abundant in the sample, and we can look here, and these are the metabolites. We come over, and these are the ones that are the most depleted in these samples. And the last thing that I'll just mention is we have a ggSave using our here package again, where we can make a PNG, plot as last plot. We can also write a delimited file type, in this case a CSV, which is going to save our plotted data just for provenance. So we run those two. Those save in our results directory. So if you run all of that, you can then come over here, results, and then you can see something like that. So that was a bit of a fire hose. But the hope would be for you then to take this code and to try to apply it to yours and then to break it and figure out how to fix it. And, I gave this to Allie, and then Allie broke it and fixed it and broke it and fixed it and eventually made the plot that she showed you guys today in the presentation, or she described to you guys today in the presentation. So with a roadmap, hopefully you can then use this to learn and to see and then say, oh, well, I don't like all of these dashed lines that you put in here. I mean, this is a log 2 fold change of a twofold, and this is 0.05 for the p-value, so I just try to sort of orient which is there. We could maybe even put in like some alpha to change the opacity or whatever. We can do a couple of different things, but the idea would be then to give you guys some code to use to try to start on the path for your own reproducible data reports. I'll stop here because it's already over time. But there's also some additional code I put here at the bottom just to explore some specific metabolites and then to use the faceting to then be able to put a lot of information on a plot that could be useful. Then lastly, I also like to print the session information for provenance and reproducibility. So you can just run this, and what you'll see is your R version, the platform, the OS, the attached packages, those are the base packages, and then the other ones that are attached that we use in this analysis. And then finally, we beep at the end because Mario's awesome. There's several different sounds you can have, and sound 8 is Mario. So, all right. Any questions? No? Maybe? Okay. Well, hey, this is class number 1, or meetup number 1. So the idea is to get your guys' feedback. I'll ask you feedback if you have suggestions to change this, make it better, make it more useful. Let me know.